So the next speaker is Dr. Bernard Bono from University of Auckland, and he is from the Auckland Bioengineering Institute, and also, as we heard before, a member of the SPARK initiative, and he's been doing really great work in bridging the museum metadata with ontologies. And we are really fortunate to have you and listen to your talk. The title of Bernard's talk is Kidney Anatomical Knowledge and Metadata from SPARK. So Bernard, please take it over. Thank you so much.
Thank you very much, Sanjay, and thank you very much, Kathy and the HuBMAP team for inviting me. I find myself unexpectedly having also an ambassadorial role in terms of bringing SPARK to HuBMAP. And it's great that we can explore synergies between the two camps. And it's both a duty, but especially it's also a pleasure. So from the point of view of SPARK, I put also a number of resources online for you to look up during this talk. Peter and I co-lead the MapCore grant within SPARK. And last year, we've also brought in Marianne Martone from ECSD to also assist us with the knowledge management, but also with the huge curatorial challenge that we have facing us. Richard Baldock is present in an advisory role and was instrumental in working with us to refine the concept of tissue domains, which I also briefly allude to. Tom Gillespie is one of the key actors in terms of knowledge engineering, and Anita Brandowski is the lead curator on the whole effort. And Natalia Kokash is the lead developer on Anatomy, which is a knowledge management tool and knowledge presentation for anatomical connectivity. And Jacqueline Bresnan is, in fact, the chair of the anatomy working group that we set up for SPARK to, in fact, provide the content expertise that we need to build the relevant ontologies to represent semantically location and much more. So in that sense, SPARK has started about 4 years ago. Over 30 groups that are actively generating data, electrophysiology that is focused, electrophysiology and neuroscience, focusing particularly on the autonomic nervous system and its relationship to visceral control. So I will start with an overview of SPARK objectives, then make some sobering considerations in terms of both data and metadata curations that would be essential to be brought center stage in the HuBMAP discussion. And then also take some considerations in terms of knowledge management challenges. So what is SPARK about? It is building a map that is a neuroanatomical map for the innervation of viscera. So both the motor part, which is the so-called autonomic nervous system, but also the general sensory for the viscera that is taking feedback back to the central nervous system. And there are a number of key constraints, and each constraint adds a new challenge. It has to be done, this map has to be done to FAIR standards. Therefore, it has to be findable, accessible, interoperable, and reusable. The navigation requires some considerations from graphical visualization, but also searchable. So metadata and ontologies are key. But also you can also be able to check out portions of this map and use it to carry out biophysical modeling and simulation. There's a huge bioengineering interest in SPARK, putting cuffs around nerves to improve conditions like bladder control, insulin secretion, cardiac rhythms, etc. Oh, by the way, we're across species. Pig, cat, mouse, and rat are primary, but there are also others. And there's slight detail about both normal physiology and disease. So there you have it. So typically our scenario is we're dealing with different groups, each producing their own map, each having their own domain. And our challenge is to make a common metaphor for these different maps and connecting them. So the notion of an independent semantic representation is key. So in fact, there are two modeling efforts that are going on in SPARK right now. One, of course, is the modeling of knowledge, which is about experiments and simulations and the data they generate. So in a sense, I expect that this is very similar also to the challenges that HuBMAP is looking at. But also there's the actual biophysical modeling of processes, of flow transfer, but also of deformation. So sometimes you can go by modeling flow in one-dimensional terms, but sometimes you need the whole vector space for 3D space to be able to operate more meaningfully. So in that sense, our challenge is to have, yes, vocabularies combined with topology to have ontologies, but the vocabularies and topologies also need to map to 3D structure. And just for you to know, Spark has a data resource center, the DRC, of which MapCore is part. And this is, in a sense, largely equivalent to the Hive module within HuBMAP. And a number of operational details in terms of the DRC have already been shared with the IU team here. So that, in a sense, by accident of history, we started about 2 years before you did. So might as well tell you about the pitfalls and the pain points that we encountered and why not.
Thank you.
And so I come to this talk with a number of preconceptions, which I may be, of course, legitimately disabused of, but I tend to look at HuBMAP as a means to classify tissue based, yes, on spatial location, that's important, but also on the basis of molecular composition. So in a sense, ontologies like Go molecular function and pathways are key to organize what emergent properties, functional properties can be assigned to tissues. And in a sense, if you look at Go molecular function, most of the terms relate to molecules interacting with each other. So molecules have to be in the same compartment to be able to bind and interact. Spark comes from a slightly different angle. Spatial location is important, but we tend to look at tissue function in terms of what route of flow, what transfer routes are being brokered by these tissues. So in that sense, it's the connectivity between one anatomical location and another. And the equivalent of a pathway, in this case, is the anatomy knowledge representation, which we'll come to. So in a sense, Spark looks at the way in which interaction between compartments is regulated, and when compartments interact, then it allows molecules to meet and fulfill their function. So in a sense, the high-level view of a vertebrate body from a Spark perspective is really captured in this simple schematic in which you have two circles and a T representation at the center. The outer circle is representing flow across the body surfaces. So this blue dot here, can I see? Can I? Oh, dash. Well, the blue dot here is the mouth. The green dot here is the anus. So this process here is the digestive tract. Here we have skin, urinary tract, reproductive tract, and pulmonary tract. The inner circle is endothelium, and that is representing, therefore, blood flow and lymphatic flow. In the center is the flow of cerebrospinal fluid. So in a sense, we tend to look at body functions as the regulation of flow, of transfers between these three cardinal flow centers, fluid centers in the body. And one of the key aspects of the map is to create semantic interoperability with 3D scaffolds of body organs that Peter Hunter will present at a later stage in this talk, with a 1D representation of the whole body on the left. So how do I move?
What you do?
Forward? Okay.
Keep the clicker as well.
Okay. So, of course, we have a natural flow for handling the data and metadata that is generated and make it amenable to mapping on the right-hand side. And I have to say one thing, if there's one thing that you remember from this talk is this, a lot of heavy lifting is really centered around data curation. That is, assembling data from 30-odd teams and for them to be prepared and organized in such a way that our curators can understand the meaning of each file and associate with each file the appropriate metadata that makes the content more searchable and more accessible. And I cannot emphasize this enough, about 80% of our effort is really individual curators within our MapCore team interacting with one identifiable data wrangler within the individual generators of the raw data. And in order to do that, we have created standards for submission and folder organization, the provision of DOIs, and most importantly of all, the provision of a free text document that is an experimental protocol that details, that accounts for the provenance and the generation of each individual file that has been uploaded. Once we have done that, we use a minimum information standard to be able to capture the metadata, and that is mapping to the original submissions. So we've created upload constraints that make sure that the curational team is not overwhelmed, and we have the right to refuse a submission if certain key conditions are not met. We have used the brain imaging data structure as an initial folder structure that we then extended to help us find files in their expected location. And the protocols themselves are semantically tagged once they have gone through protocols.io, in which the data curator and the data wrangler go through the actual protocol and organize it in such a way that it makes it much more meaningful to understand. There are over 5 full-time equivalents on the data and metadata curation team, and I have to say we are already stretched. So this is something that you need to bear foremost in my mind. This is our dashboard, a Google spreadsheet that allows us to keep track of the curatorial stage and the pitfalls and the errors that they've encountered with each team. From a metadata perspective, we've created a working group that collects essentially the requirements for metadata across the various domains. Essentially, we went to each team and asked them the question, how do you expect to search the data, both your own data, but also other people's data? And we collected these requirements into a spreadsheet. We've gone through a couple of iterations of feedback and then created a class set that allows us to assemble Lego style. Why am I going back? Oh. Assemble Lego style in OWL the various aspects that account for an experiment. And to give an example, we have a set of files that have been uploaded to our repository. And the experiment class is telling us that there was electrophysiology carried out in this location in the heart. And here is the background hemodynamics for the, in this case, the pig during the acquisition. Here's the raw electrophysiology, and here's the post-processing. And this is the analysis we carried out. But then this experiment triggered another experiment. So we have a whole narrative in the metadata that is taking us from an initial experiment and then corollary experiments that were carried out. So here we have the actual histology, and then from the histology there was RNA-seq acquisition. So then, of course, we need to create a map that makes sense, that organizes and classifies the metadata associated with the data that we've collected. And in order to do that, of course, we need to map to a core set of ontologies that provide meaning, both in terms of the minimum information standard that we have collected, but critically, so that communities outside Spark are able to use the same ontologies to be able to find us. So we add value to our data by making it findable by others, and we of course can leverage other people's resources as well. And therefore, this is a question of cohesion internally, but also adhesion with the rest of the biomedical community. The choice that we went for was for the Interlex set of ontologies that comes with a considerable infrastructure associated with it. We're also leveraging research resource identifiers, RIDs, for antibodies, for methodologies, for plasmids, etc., as well as, of course, orchids for people. And the infrastructure that we leverage is the UCSD SciCrunch, and there are a number of technical details that I can share with you later. But at a very high level, we take the minimum information standard on OWL representation and map it to the Interlex ontologies. And the protocol, as well as the bids submission, is then semantically tagged semi-automatically to create a mapping between various files and protocols to the core ontologies that we use. And for, I'm pressing things that I shouldn't be. And this makes it available to also search from the outside. So using SciCrunch, people coming from, say, neuroscience or cell modeling can also find Spark resources once these are released. And for the subset of ontologies that are focusing on anatomical location, we have created tools such as Anatomy to create a visual projection of the anatomical knowledge to also help create a kind of underground tube map representation of body connections to be able to then put metadata points associated with data that is germane to those locations. And as you saw, these anatomy maps are also interoperable with 3D scaffolds that Peter will present. So the last 2 minutes will be focusing really on anatomy and its transition. So anatomy started out as a visual map, as a tree map that takes ontologies such as the FMA and creates a visual constraints that puts surface organs structures on the outside of an island and the inside structures, the inside tiles are representing internal connections, and we use to use Anatomy tree maps to overlay protein structure, connectivity from, say, cardiovascular system, as well as neural structures. But what immediately became clear that it was creating some confusion in terms of putting nodes on top of tiles that seemed to suggest that a particular connection was passing through a region that had absolutely nothing to do with it. So, and this is where that was done 6 years ago. Since then, our thinking has evolved. We want to create a much more process view of the body, and that led us to start thinking in terms of conduits. Our initial thoughts about conduits started work with Richard Baldock, where we talked about what is the prototypical tissue domain, which we refer to as a primary functional tissue unit. And the notion of the primary functional tissue unit is in fact a backbone, like a protein domain, but the backbone here is an advective tube that could be either a blood vessel or an epithelial tube. And these cuffs are really guaranteeing that any 2 points within that cuff is within diffusion distance of each other, but that cuff itself is centered upon a long-range tubing that connects to either the endothelial or epithelial tubing. So in that sense, a lot of physiology goes on in regulating the interaction between the overlap of a diffusive domain for long-range advective tubing by regulating what molecules can be ferried from one system to another. And this led, therefore, to the concept, I'd like to move on. This led to the concept that we can generalize upon the notion of a conduit that can be applied to different systems. Essentially, many of the organs are representing an arborization of conduits at different scales. So here on the left, we have the lung. Here on the right, we have, of course, the nephron, but also the membrane channels within the cells are themselves conduits. So we created a scale-free methodology, a grammar, if you want, of conduits that have different topological conformations that can be assembled Lego style to represent any kind of advective system. So here we have a caricature representation of the entirety of the urinary tract. On the left, we have a representation of the ureteric meatus. On the far right, we have the Bowman's capsule, and we have now a topological layered representation of the key anatomical structures that line the wall for the urinary tract that are then mapped to the various ontologies. So essentially, we view the body, the vertebral body, as a set of arborizations that can be modeled canonically using anatomy and grammar. And probabilities of branching, thickness, and length can be associated with these conduits, and then they can be used as an emission, in this case, to create a lung representation that has been emitted from a canonical representation of anatomy for the airway tract. And this is work that is ongoing to create a representation, in this case, of the kidney, where we can find 3 types of arborization, epithelial arborization, and 2 cardiovascular arborizations that link up in the urinary kidney lobus. And each edge is associated with a conduit that accounts for the flow that is conveyed. So in that sense, anatomy is used as one way to capture in a very succinct and terse way the representation of key conduits around the body for the epithelial system, the endothelial system, as well as, of course, neural connectivity. And this is one final slide that reminds us that the same type of grammar can be used also to capture membrane channels conveying, in this case, sodium across plasma membrane and to the sarcoplasmic reticulum. This is my final slide. Essentially now, anatomy is being used to demonstrate semantic connectivity between different parts of the brain, but passing through other organs. So starting with the nucleus tractus solitarius, we can reach the superior cervical ganglion because of connections that have been captured in anatomy passing through the larynx. So this allows us then to use semantic search over connectivity models to find data that is coming from across connectivity maps in the brain. And with that, I thank everybody and open to questions. Thank you.
Thank you so much. Yeah, we have a few minutes for questions. That was a great presentation. So please go ahead with questions.
Okay. Not so far. Okay.
Well, I'll start with one. So Bernard, I mean, it was impressive. You have 5 full-time equivalents to really curate the metadata. So how often do you find discrepancies, you know, and how do you handle them from in your curation of the metadata coming from the different sites?
By default, great question. By default, there will always be discrepancies. We're surprised that there aren't any. So in that sense, it is key that we have a curator assigned with a single point of contact for each of the groups. And there is, first of all, a report that is built, and there is a key understanding of the meaning of what the data wrangler says about the metadata that they have completed, and the interpretation of the curator of that metadata has been sent to us. Bear in mind that one key way that we have used to guarantee that there's homogeneity in terms of quality of metadata is to have a curator who's well-trained and then goes out and speaks to people and then uses their interpretation to alter the metadata themselves. It is tempting to get people to fill in their own metadata forms, understand it and use that, but there's so much heterogeneity in understanding and so much training that is required. And people, let's face it, people filling in metadata is not considered the highest priority in it. Okay, you need to have professional curators who sit in the next desk with the knowledge engineer and the ontologist who can help with making the right choices and creating the right type of representation. So always assume there's going to be discrepancy in the metadata. If you're not finding any, you're not looking hard enough.
Yeah, thank you. I think those are really great points, important points to remember in any activity of dealing with these kind of information collection. Any other questions?
I want to follow on that. How much of the, you know, it's the sort of 80-20 kind of estimate of that activity that you're describing of the curation is translation into metadata that those 5 people have come up with a common understanding of? So how much of it is that translation, and how much of it is going back to collect things in a timely fashion that were just left out?
The latter.
So lots of the latter.
Absolutely the latter. It's,
Okay.
it's the informatics practice. It's finding the files. It's also chasing people.
Yeah.
This is a bottomless pit,
Yeah.
which is why we had to create constraints. We put up a contract. We said, we'll look at the stuff that you upload only if you satisfy these constraints. We had to retain a certain amount of control because curators can be easily overwhelmed. And dealing with 30 groups has an economy of scale that can be quite terrifying in a certain respect.
So really, it's that part of it is more about the collection than the synonymy. By centralizing it,
yes,
you can manage the synonymy, but the reaching out part and the big activity is all the collection itself.
I was sitting there when you were talking about.
That's very useful.
I was sitting there when you were talking about what ontologies to use, and part of me was saying,
Doesn't matter.
you wait. You just wait. It's nice to talk about this, but you are lucky to have any mapping at all. Okay, it is that bad, and I'm saying it while cheering you on because we have to do it. But
Thank you.
yeah.
We appreciate that.